So why don't you go ahead and get started?
Sure. So hi everyone, I'm Andy from the Hancock Lab, and today I'll talk about the role of S100A9 in cellular reprogramming and sepsis. Next slide, please. So sepsis is usually referred to as a bloodstream or systemic infection, but it's officially defined as a life-threatening organ dysfunction caused by a dysregulated host response to infection. One aspect of the dysregulated host response is a phenomenon called cellular reprogramming, or CR, a form of immune paralysis induced in immune cells like macrophages after repeated stimulation. For example, in the top row, one stimulation of lipopolysaccharide, or LPS, which is a bacterial cell wall component, can make the macrophage produce pro-inflammatory cytokines like TNF-alpha, but a second stimulation with LPS does not induce any release in TNF-alpha, indicating it's reprogrammed. So our lab found 99 genes that were differentially expressed in CR, which we call the CR gene signature. And this gene signature was able to predict if ER patients had sepsis or will undergo organ failure. So we hypothesized that a similar two-hit model is occurring in sepsis patients. So on the bottom row, you can see an initial preclinical infection like a pneumonia either spreads systemically throughout the body, or say the patient gets another infection in the hospital, and this leads to sepsis and detection of CR. So understanding the mechanisms of CR is really important for understanding the immune response in sepsis. Next slide, please. So what I was interested in is a calcium binding protein called S100A9, which is actually one of the genes in our gene signature. So a bit about it, it's released during inflammation by macrophages that can start inflammatory signaling pathways and is considered a damage-associated molecular pattern. There's also increased gene expression of S100A9 in sepsis patients with worse outcomes, as you can see on the graph on the bottom left. And when we create a protein-protein interaction network from all the genes in our cellular reprogramming gene signature, S100A9, which is circled there, has many interactions, making it a hub gene and potentially a key regulator of CR. So I hypothesized that S100A9 is required for cellular reprogramming induction in macrophages. Next slide, please. So first, I stimulated THP1 macrophages with recombinant human S100A9 protein, followed by LPS, and compared it to macrophages exposed to either 1 or 2 doses of LPS. So on the left bottom graph, as expected, the single LPS dose elicited the release of the pro-inflammatory cytokine TNF-alpha, while very little TNF-alpha was released in the double stimulated or reprogrammed macrophage. And the S100A9 treated macrophages also had significantly lower release of TNF-alpha and resembled the cellular reprogramming condition, suggesting that S100A9 was able to induce CR. I also measured IL-10, an anti-inflammatory cytokine, but unfortunately there were no significant differences between the conditions. Next slide, please. So if S100A9 induced cellular reprogramming, I then wondered if inhibiting or removing S100A9 could prevent CR. So on the left, I first used an S100A9 inhibitor called tosquitamod and added it with LPS to THP1 macrophages to see if the dampening of the TNF-alpha response could be prevented. However, the difference was not statistically significant despite trending towards prevention. And IL-10 also had no significant differences. And then on the right, I then used CRISPR-Cas9 to mutate the S100A9 gene in induced pluripotent stem cell derived macrophages to create a truncated S100A9 protein. But again, the mutant macrophage was still able to become reprogrammed. So overall, neither inhibiting nor mutating S100A9 affected cellular reprogramming induction. Next slide, please. So to conclude, I was interested in S100A9 because it's a hub gene in our sepsis predictive cellular reprogramming gene signature, and also highly expressed in sepsis patients, making it a potential target for preventing cellular reprogramming. But while adding S100A9 could induce CR, removing it from the system by inhibitors or through CRISPR-Cas9 unfortunately did not prevent CR. So I conclude that S100A9 is likely involved, but not required for cellular reprogramming. So my next steps are to further characterize these mutant macrophages by profiling other cytokines, performing flow cytometry, or functional assays like phagocytosis and wound healing assays, as well as investigate the role of other S100 proteins in cellular reprogramming and sepsis. So thank you for listening, and I'm open to any questions from the audience.
Great. Thanks very much, Andy. So questions can go either in the chat box or raise your hand. And I'll stop sharing so we can get the Hollywood squares back.
Hello.
Hi, Dr. Nee, go right ahead.
Yes, so I found it's pretty quiet. So what the macrophage you used here? So whether that's the monocytes or some tissue macrophage or some special tissue, for example, Kupffer cell from the liver will be different from other results of macrophage.
Yeah, so
during the sepsis. Yep.
Right, the S100A9, the recombinant human S100A9 protein and the tosquinamod experiments were done with the THP1 monocytic cell line. So these are blood monocytes stimulated or matured into macrophages. I think they're a leukemia cell line. And then the mutant CRISPR-Cas9 macrophages, those are induced pluripotent stem cell derived macrophages. So there's, again, blood, it's supposed to resemble blood monocytes that are stimulated to become macrophages. And we've compared them to human primary monocytes, and they're quite similar.
Thank you.
Thanks.
Any other questions? Well, if not, thank you very much, Andy.
Thanks.